Good afternoon and welcome to the penultimate webinar in our 2021 webinar series delivered to you by Dr. Tim Sandle. Our monthly webinars have been covering different aspects of Annex 1 to hopefully help you prepare when it goes live early next year. Today's topic will cover sterility assurance in cell therapy products. Before we start the webinar, I'd just like to do some housekeeping with you. If you have any technical issues, if you'd like to send a short message in the questions box on the right-hand side of your screen, we'll try to answer it. During the webinar, we'd like to ask you to ask as many questions as you would like, also using the question box on the right-hand side. At the end of the webinar, time permitting, we'll try to answer as many of these as possible. Any we don't get around to, we will answer offline, and I will share the answers with you along with a copy of the slides from today's webinar and a link to the webinar itself. So, welcome. If you are joining us for the first time, my name's Annette Russell and I am the Sterile Manufacturing Lead at RSSL. I work in the commercial team at RSSL, but I work directly with our pharmaceutical microbiology team, who are there to support our clients who are involved in both sterile and non-sterile manufacture. To those who've been to some of our previous webinars, welcome back and thank you for our support. And we hope that we can help you going forward. So, as you can see, my contact details are on the slide in front of you. If during the or after the webinar you have any questions or you would like to know more about RSSL's capabilities, please feel free to contact me. So this webinar, I'm continuing our Meet the Team. So I'm going to talk to Thomas Gosling, who is one of our senior scientists in our microbiology team. Hi, Thomas.
Hi, Annette.
So, Thomas has worked in our pharmaceutical microbiology team at RSSL for more than 7 years and has experience of many of the different aspects of our microbiology services. He's previously managed the endotoxin analysis before recently moving into the sterility when we opened our new facility. So, Thomas, can you give us a brief overview of what your role at RSSL is?
Sure. So I'm currently based in our sterility team, where I perform a lot of testing using our Grade A or ISO 5 isolators, and they're located in our Grade D clean room. I assist in the organisation of the laboratory and its workload to ensure our client timelines are adhered to. I also do a lot of the sterility validations. This can encompass everything from a simple method transfer all the way to troubleshooting complicated or new products. I create the protocols and methods and create these into finalized testing reports.
Excellent. So what does a normal week look like for you at RSSL?
So it begins with setting up the isolators for use and scheduling the workload. The role is heavily laboratory based with testing and releasing products, and this is broken up with client calls and updating processes and things like this as well. The data dynamic and variable keeps us on our toes, and we have to deal with a fluctuating workload and also the technical problems that can arise when dealing with finely tuned piece of equipment such as isolators.
So you mentioned that you are involved in running and writing the sterility validation for our clients, and this has included some of our cell and gene therapy clients. Can you tell me what the main challenges you've come across with this particular type of work?
Sure. So there are a number of problems that can appear when doing sterility testing and validations. They can appear when a product has antimicrobial or antifungal properties. This will affect the growth of the specific organisms that you need to add during validation. We can overcome this by using specific kit types, increasing or altering the rinse fluid amounts, and this is usually enough to overcome the problems. However, sometimes we may have to use specific rinse mediums, chemicals, or enzymes to inactivate the sample components. The preferred pharmaceutical method is membrane filtration, but due to increase in cell-based samples that are too large to be filtered, we're increasing looking into direct inoculation to overcome this problem. And then also rapid sterility methods are also being introduced as well, which may overcome some of these problems.
Okay, excellent. And so finally, what do you choose to do for downtime when you're not involved in all the sterility work at RSSL?
So outside of work, I have a 5-month-old daughter and a rescue dog, and they obviously take up quite a lot of my time. Outside of that, I play hockey, search out new music, and then I'm also continuing a house renovation, which is over 3 years, but we're almost there. So that takes up most of my time.
That sounds quite ambitious with a young baby and a rescue dog.
Of course.
Okay, thanks, Thomas. Thanks for
Thank you
coming
very
on
much.
board. So, as I mentioned, at RSSL, we can help support our cell and gene therapy clients in several different ways. So, we can help with your characterization of your product with bespoke bioassays, as well as kit-based bioassays. And we have extensive experience of running ELISAs and Western blots, SDS page, and immunoassays. We also do an awful lot of raw material testing, and we can help with those non-compendial, often more difficult specialized raw materials that are used in this sector. We have an extensive method development and validation capabilities that we can use to help you in these testings. As you are aware, at RSSL we have the microbiology team that can help with the sterility, the bioburden, the endotoxin, and we can also help with other elements of safety testing, such as E&L profiles of any infusion bags that you may be using, or help detect or troubleshoot any impurities that you may find occurring in your product. And finally, we can help support your sterile facility with cleaning validation, air and water monitoring, or with our training group, we can do some specialized training specifically for you in this sector. And this is all in line with the Annex 1 support that I've talked about over the preceding webinars, so we can help you in a variety of different ways. So that's enough of me now. I'd like to take the time to introduce our guest speaker, Dr. Tim Sandle. I'm sure many of you have heard of Tim and subscribed to his many publications. Tim has over 25 years of experience of microbiological research and biopharmaceutical processing. Tim is a member of several editorial boards and has written over 600 book chapters, peer-reviewed papers, and technical articles relating to microbiology. Tim works for a pharmaceutical manufacturer in the UK and also finds time to be a visiting tutor at both the University of Manchester and UCL. Hi, Tim.
Hello, thank you very much, and it's a pleasure to be with you. Right, I'm just going to share my screen. Okay, does that look okay?
We're seeing your notes as well, Tim.
Okay, how's that?
That's brilliant, thank you.
Brilliant. Okay, thank you very much. Well, it's a great pleasure to be with you, and we're going to be looking at an exciting emerging field of medicine. So this is the variety of cell therapy products. So as some people are going to be a little bit less familiar with these products, we'll just talk about them a little bit and then cover some of the general challenges, and then we'll have a look at some of the sterility assurance concerns. We'll have a look at sterility testing, and particularly in either circumstances where that may not be possible or where we may want to look at rapid methods. And we'll also touch a little bit on mycoplasmas, which will feed in nicely into the next webinar. Okay, so we've got this kind of broad collection of emerging products, which we can put under the banner of advanced therapy medicinal products. So this is a combination of gene therapy medicinal products, somatic cell therapy medicinal products, and tissue engineered products. And these represent truly groundbreaking and new opportunities for the treatment of various diseases, and they're particularly important for severe, previously deemed to be untreatable or chronic diseases, for where conventional approaches in medicine have proven to be inadequate. And there's also a specific category as well we can add, which is where these advanced products are combined with medical devices. So this is what comes under the combination product banner as well. So just a little bit of history. The first ATMP product approved in the European Union was back in 2009. And this was with the authorization of a product called Chondro Select, which was a tissue engineered product. And it was developed for the treatment of cartilage defects. And with this particular case, during osteoarthritis, where there's a degenerative effect happening, then major modifications to cartilage are observed at the sort of tissue and cellular levels. And this can lead also to lesions as well. So this was a pharmacological treatment based around tissue engineering, and was a step towards regenerative medicine designed to induce cartilage repair. In the US, the ATMP first registered there came out around 1 year later, and that was called Provengene. And that was somatic cell therapy treatment for prostate cancers. Now, just looking at these different types of products in a little bit more detail. So we have gene therapy medicinal products, and these are products containing an active substance which contains or consists of a recombinant nucleic acid. Then with the somatic cell therapy medicinal products, these are about the clinical application of cell therapies and tissue engineered products, and this falls under this banner of regenerative medicine. So that goes back to that first European Union product. Then there are products that consist of engineered cells or tissues. And there are some examples of different product types as shown on the slide there. So we have those based around mRNA, which has obviously been in the news quite a bit with some of the coronavirus vaccines. The use of a non-viral vector, what might be called naked DNA. Then products that use viruses to introduce the active drug product, and also genetically modified cells. And in terms of this sort of general banner of advanced, this must incorporate as an integral part of the product one or more medical devices. It needs to be cellular or tissue containing living cells or tissues. And these need to be able to act upon the human body with an action that is primary, that has a direct action towards the disease that it's supposed to be treating. Now, as pharmaceutical products, then all ATMPs are classed as a type of biologic. And they also form within this personalized medicine or precision medicine narrative that's evolving as well. So this is the philosophy where parts of pharmaceutical manufacturing is gravitating away from the centralized or generic manufacture to a model where medicines are brought closer to the point of care and are developed for individual patients or smaller groups of patients. Beyond that, definitions of human cell-based medicines are difficult because they're fairly heterogeneous kind of collection of medicines varying in the types and origins of the cells and their relative complexities as well. And also the cells could be originating from the same patient, or they could be allogeneic, which is from another person going into the patient, and then some others are also genetically modified. So you can get a sense of the complexities that are around that. Another sort of general challenge is the cost, because the development costs are very, very high, and this can run into $1 million a patient in many cases. So the development requires high upfront costs, which makes it challenging sometimes for new entrants into this emerging market, and also is offset by the affordability of sometimes the relative treatments. There's also complexity sometimes around bioethics, particularly in place for genetic research and collection and storage, and even in terms of ownership of material, the necessity to get informed consent, patient confidentiality issues, the security of biobanks, and so on. Plus, there's the scientific complexity that we've discussed, and then the central point to this webinar is around sterility assurance issues. There are also specific GMPs, and there was a debate in industry about whether or not there should be separate standards for ATMPs, and the regulators both in Europe and the US decided that was appropriate because these are complex products, and it follows that the risks may differ according to the type of product, its nature, characteristics, particularly the starting materials, and the level of complexity within the manufacturing process. It can also arise that the finished product should have some degree of variability as well because of their biological materials after all. So that also needs to be taken into account. So within Europe, the core GMPs are displayed on the slide, and it was on the 22nd of May 2018 that the primary GMP annexes came into force, and we'll touch on some aspects of those when we look at any of the specific sterility assurance issues. In the US as well, then there are equally similar emergence of guidelines as well. And as we know from previous webinars, the FDA guidelines tend to emerge more quickly and more commonly, or more wide-ranging than they do in Europe. We also need to take note of ICH Q5D, which is about the derivation and characterization of cell substrates used for production of biotechnological or biological products. And also ICH Q5A, which is the guideline on the quality of biotechnological products in terms of viral safety for products derived from cell lines of human or animal origin. So there's a raft of emerging regulations also that need to be navigated through. There's also, as with all aspects of pharmaceuticals, but again, within these GMPs, there's specific reference made to quality risk management. So there's that requirement that a risk-based approach is adopted to enable the manufacturer of these products, the design, the necessary organizational, technical and structural measures that are required to meet GMP expectations. And these will need to be tailored to the specific risks of the product and the variations and complexities of multiple steps within the manufacturing process. So there's a specific reference there that some of the general aspects of risk assessment will apply, but equally there will be some special areas as well. So this requires a degree of flexibility, as well as a need to ensure that risks have been appropriately mitigated and there's suitable controls in place to assess any residual risks that might remain. And such a task is going to be made easier with a data-driven and scientific approach. And a lot of the risks in relation to ATMPs is dependent upon the biological characteristics and the points of origin of the cells and tissues, particularly protecting them from viral or mycoplasma contamination. But account also needs to be taken of the biological characteristics of the vectors. So this could be transgenes or reverse transcription. And further risk factors stem from the level and characteristics of the expressed protein in gene therapy products, or the properties of other non-cellular products that could be from specific raw materials or from different matrices that are needed, plus the actual manufacturing process and the degrees of controls around that. And where the sterility assurance aspect starts to emerge is with production processes that require particular complex and substantial manipulations, which are theoretically going to be at greater risk than less complex processes. And a further risk factor, as we know from, again, from previous webinars, the longer a given process stage, then the greater the potential for microbial contamination to occur. And again, with many ATMP manufacturing processes, because the processes will often necessitate a long exposure of cells or tissues, then those possibilities potentially become greater. And in terms of these sorts of controls, it's good practice that the development program tries to address some of the risks. So in terms of choices, material and types of processes that are there to be scaled up into main production. And these should be addressed from the point of view of the biological or therapeutic function, the maintenance and the protection of the cell population. And also the risk based around the administration of the cell-based medicinal product is also important. And that will also relate to the formulation and method of delivery. And at this stage, there are some aspects of the design, the quality by design aspects to go into the process. So we want to ensure we have absence of components or materials that might create leachables that then could be toxic to cell growth or impair the intended performance of the medicine. And also to have a good understanding of things like surface chemistry and strength to ensure that we're getting good optimization of cellular growth and do what we can to avoid contamination. Okay, so moving on to some more sterility assurance specifics. Always at the top of any sterility assurance concerns comes people. So it is important that people are adequately trained, and in particular personnel working in clean areas should have a good grounding in aseptic manufacturing, and particularly with smaller startup companies, then it's not necessarily always the case that this knowledge is there or the people involved are always as well grounded. So having an understanding of the basic aspects of contamination control and microbiology is important. And training should include adequate gowning requirements, an assessment of the competency of the personnel to work with aseptic production areas. So this will involve annual say broth transfer tests and annual assessments of good gowning practices. Special training should be in place to prevent the transfer of communicable diseases from biological raw and starting materials, both from the operator to the materials and often vice versa as well. And personnel who are handling genetically modified organisms should have additional training. So they need to be cognizant with cross-contamination risks and also potential environmental impact should any GMO be released into the environment and the safeguards around that. And also there should be clear allocation of responsibilities, which sits with any good pharmaceutical manufacturing arrangement. Then these concepts should also extend to the strict controls in place for the movement of people. So for example, avoiding personnel or with the transfer of items from passing directly from areas where there is exposure of live microorganisms or genetically modified organisms, toxins or animals into areas where other products are being prepared or products that have already been inactivated or where different organisms are being handled. So this requires a detailed biosafety risk assessment along with appropriate control measures and should be supported with aspects of clean room design and with good cleaning and disinfection practices as well. So this premises aspect also extends as well to other factors that are going to keep the facility in a GMP state. So it's important that the production processes are clearly understood, that we have appropriate records in place, that we have a documented process flow. So for example, on the slide there is a generic gene therapy process flow chart. Of course, it's very generalized, but each one of those steps needs to be understood and controlled with appropriate specifications laid down for the materials, intermediates, bulk products, finished product, and so on. And also validated to ensure consistent production and to make sure that the quality control system and the staff engaged with quality control are operationally independent from production, so they can ensure that standards are enforced and to address deviations, change controls and CAPAs and so on under a separate system so there's no risk of interference. Further with the design of premises, it's important that the environment is protected. So production should be taking place in areas connected in a logical order with a corresponding sequence of operations and a cascade of required levels of cleanliness. The handling of ATMPs containing genetically modified organisms does, as I said earlier, pose a risk to the environment. So this requires the implementation of additional quality by design principles when the facility layout is considered. And this is from both a point of view of avoiding unintended release of materials and also in ensuring the sterility assurance of the product as well. So designing quality up front and incorporating quality into the initial design process to look at the requirements for the environment, equipment, components, materials, processes and personnel is each an important factor. Plus having the risk assessments in place to prevent the introduction of wild type viruses and viral vectors and ensuring that things like adequate biological safety cabinets and adequate air control and airflow are in place. So further with this sort of minimising the risk of cross-contamination, it should stand that the concurrent production of two different ATMPs in the same area is not acceptable. The only time this could be considered is when dispensing, where there may be two different isolators operating. But even here, good consideration needs to be given to the room airflow dynamics and ensuring things like 100% air exhaustion from the room and from the facility. So recirculated air would present a danger. And across the whole facility, it's important that the air handling units are designed, constructed and maintained in order to prevent or to avoid the cross-contamination occurring between different areas of the manufacturing system. And also having a positive pressure cascade. So the airflow is from areas of higher cleanliness to adjacent less clean areas, apart from where negative pressure might be required for specific areas for reasons of containment. So for example, that could be around the replication of competent vectors or where pathogenic bacteria are used. And in these cases, the negative areas should be surrounded by positive pressure clean areas of the appropriate grade, so the contamination is kept within. Further important measures include before any manufacturing operation starts, steps should be taken to ensure that the work area and equipment are clean and disinfected and free from any starting materials, product residues, or anything else. That might be documents or unwanted materials not required for the current operation. It's especially important that mix-up of materials is prevented, and special precautions should be taken to avoid mixing human-derived cell materials or other dedicated materials. Every stage of production, products and materials should be protected from microbial and other forms of contamination. And also, these measures should extend to the preparation of solutions, buffers, and any other component that needs to be added to the process in order to, again, protect the product from contamination. And the general cleanliness levels should extend to low microbial recoveries throughout the clean areas in which the production's taking place. Furthermore, in all manufacturing areas, any step that might lead to the unwanted formation of aerosols, such as from centrifugation or working under vacuum or the homogenization or sonication of a solution, then appropriate mitigation measures should be implemented again to avoid cross-contamination. And special precaution should also be taken when working with infectious materials, and measures to prevent cross-contamination should always be risk-based. And that loops back to what we were looking at earlier. And further with the concept of segregation, then there are different measures that can be taken. So this could include dedicating the whole manufacturing facility or a self-contained production area to specific types of cellular products on a campaign basis, so that different products are separated by time with a thorough decontamination step in between. Greater assurance can be produced through the use of closed systems for processing and material and product transfer between equipment. Any accidental spillages, especially of live organisms, need to be addressed quickly and safely. The use of airlocks and pressure cascades, as we mentioned, to confine potential airborne contaminant must be engineered in. Where possible, single-use disposable technologies should be used. The cleaning and disinfection procedures should be adapted to the special characteristics of the product and the manufacturing process. There should be documented and assessed cleaning and decontamination steps between every batch, and this might vary going back to that campaign thing to be at different levels, but it certainly needs to be something thorough in place. And other suitable technical measures need to be considered, so like dedicated filters and so on. Other suitable measures include keeping specific protective clothing inside areas where products with a high risk of contamination are processed, implementing adequate measures for handling waste, having methods in place to collect and dispose of contaminated rinsing water and soiled gowning, and also keeping with those restrictions on personnel that we discussed earlier. Now, looking at the closed systems advantages a little bit further, in closed systems, then theoretically there's no exposure of the process to the surrounding environment. Closed processing prevents the ingress of microorganisms from the environment, and it incorporates single-use disposable components for any material that comes into contact with the product, which can then be decontaminated and destroyed. All components and reagents should be pre-sterilized using filter sterilization. And this approach, this kind of level of robust control enables processing of multiple patient batches running consecutively. But a lot needs to be placed onto the decontamination methods, and a good disinfectant is required. So often this necessitates the frequent application of a sporicidal agent, and if there were risks of prions, for example, then and any equipment needs to be reused, such as a stainless steel component, then the CDC guidance for prion inactivation would necessitate the use of 1 molar sodium hydroxide, followed by autoclaving at 134 degrees Celsius for 30 minutes. So you get an idea of that level of control that's required if there was a prion risk. In terms of general cleaning and disinfection, then there is also the now increasing frequent use of vapor hydrogen peroxide products as well. Other key sterility assurance aspects include controlling of repairs and maintenance, particularly if anything needs to take place during the manufacturing operation. It's important that these activities do not present any hazard to the quality of the products, that the premises have all the measures in place to avoid against the entry of pests, particularly things like insects, that there's nothing toxic manufactured or used anywhere near the processing areas, so be a complete avoidance of any pesticides or herbicides, and that anything that comes into contact with the product has been adequately assessed, that there's no reactive materials that might contaminate the cells or render anything non-sterile. There should also be bioburden testing of key intermediates and a program of in-process controls and in-process testing. And these tests can be used to generate some key critical quality attributes and give an indication of the likely contamination of the finished product. So there should be bioburden determinations, particularly at the point of ingredient additions and after each hold stage, because that is the point where microbial growth can commence from. There also needs to be a good environmental monitoring program in place, and that should form part of the contamination control strategy. And that should include physical and microbial assessments, so that will be viable contamination and an assessment of inert particles, plus the extension that the physical controls are working, so an assessment of things like air pressure differentials as well. And the standard array of methods should be used, so surface, air and personnel monitoring, using a risk-based approach for selecting of environmental monitoring locations. And that becomes particularly important if, say, the product needs to be used before a sterility test can be, a sterility test result is known, because having a thorough understanding of the potential environmental risks is important, and the only meaningful way to do that is through a sufficient number of samples at a sufficient frequency and subjecting that to trend analysis. Environmental monitoring extends to the necessity to monitor the grade A area continuously, with the only caveats being that unless there was something that might damage a particle counter through a high particulate generation, or would present a particular hazard if live pathogenic organisms are being used. Otherwise, the same standard as applied to any other sterile product should be in force. And that would include also the monitoring of personnel working in the aseptic areas, particularly after performing critical operations and prior to leaving the area in the form of finger and gown plates. And there should be a system in place to disqualify personnel with problematic trends, and a system in place to requalify personnel. And that may include participation in a media simulation study. And the media simulation study also represents an important part of the environmental controls as well, because it will assess the robustness of the process and all of the steps involved to ensure that the product can be filled under controlled conditions. Also need to be mindful of impurities, because during manufacturing, variable amounts of impurities, both product and process related, could potentially be introduced into the final product. So it's crucial that any reagents known to be harmful to humans are analysed, and acceptance criteria set. And the presence of impurities extends to adventitious microbial agents, so it could be viruses, mycoplasma, bacteria and fungi. And this form of contamination could originate from the starting raw materials or be adventitiously introduced during the manufacturing process. So again, a risk assessment would be required to evaluate the possibility of these agents getting in, or their potential reactivation through the process. So a lot of aspects of the in-process monitoring will need to ensure the absence of bacteria and fungi, any particular viruses of concern, and mycoplasmas. And with mycoplasmas, which will be the subject of the next webinar, this is again an important control measure, particularly if there was high levels of microbial contamination, in particular mycoplasma contamination, then this could have an adverse effect on the morbidity and mortality of the recipients of the product. And another important area, so come on to mycoplasma in a second, is with the importance around the actual sterility test as well. So one of the concerns is that a number of products need to be used in advance of the sterility test. So either that's something that has to be accepted then reacted to, or there are the emergent rapid microbiological methods. But in some cases there are questions around the accuracy of rapid methods as well, and certainly in degree of the level of detection. So there is some controversial discussions around infectious dose. So this is based on the philosophy that if a rapid method cannot always detect down to a single colony unit, to what extent does that matter if there's little evidence of one colony unit in the product being clinically significant? And this is a debate that's ongoing in parts of the US. At this stage, the requirement for sterility is the requirement for sterility. So my advice would be not to go down that route unless there was specific regulatory direction. So just looping onto mycoplasmas specifically. So mycoplasmas are a form of bacteria that can infect cell cultures, and they are a potential threat to either the manufacture or release of the product or to the recipient of the cell therapy product. And there's a number of conventional culture-based mycoplasma methods supported by staining that are highly sensitive, but they require considerable time to obtain the results. They could sometimes be up to 28 days. So in many ways, the culture-based mycoplasma tests are not necessarily going to fit very well in the production of these next generation products where there might be an urgent need to produce something for a very ill patient. PCR-based mycoplasma testing is now relatively robust and is a much faster way of making the assessment. But of course, the methods need to undergo strict validation, and it's important that the each sample type is assessed against the assay as well. Again, just touching on the sterility test, sterility assurance aspects, again, because of the weaknesses around with the sterility test in terms of numbers of samples, in terms of recovery of stressed organisms around incubation time, then this connects with the whole Annex 1 strategy of a robust sterility assurance system and drawing more inferences from that totality of data rather than over dependence upon a single release test. Alternative sterility tests are suitable for some types of ATMP cell-based products, such as blood culture systems, and these provide automated and continuous monitoring with the objective to detect microbial growth. A complexity that arises here is with sometimes the already turbid nature of some cell therapy products, which produces difficulties with culture interpretation when either aerobic or anaerobic enriched media is used. So this is why some are going down the ATP bioluminescence technology route or with carbon dioxide monitoring systems as alternative methods to assess the likelihood of any microorganisms being present in the finished product. A further test that is important, and it's one of the prerequisites for sterile products, which is for them to be free of particulate matter. And as cells in suspension are not clear solutions, then the GMPs permit the replacement of the particulate matter test by a different appearance test. So it could, for example, be an assessment of product colour. So the strict requirements for free of visible particulates, this is an exception within the GMPs, but this is provided that alternative measures are put in place to control particles from materials. So this might include, for example, the filtration of raw material solutions, and with other controls around manufacturing. An alternative approach is to simulate the manufacturing process with an alternative, a placebo or something like that, so a material without cells, and then do a test to see if there's any particulates present. Another important control is also with the absence of pyrogens as well. So this could be the test for bacterial endotoxins, or it could be the need to expand that net further with a wider test for pyrogens, as with the monocyte activation test. Okay, so bringing this webinar to a close, there was obviously a lot of things to cover, and given that these products are very specific, I could only really address the subjects in sort of very general terms. But we have looked at the emergence of these patient-specific therapies and some of the new challenges they present, and the way that regulators are driving a special wave of GMPs, and that's an acknowledgement that the sterility test may not always be available. So we're much more reliant upon other control measures and modifications around things like visible particulates as well. Okay, so thank you very much, and I'll pass you back to Annette.
Thank you, Tim. I'm just going to take back presentation. So I have some questions that have come through. I will try and get as many answered as I mentioned. So the first one I have was from Eugene, and he said, are the regulatory requirements for visible and subvisible particles in ATMPs? And if so, how do you deal with it? I guess it's hard there.
Yeah, so there is leeway if it can be shown that the products have a natural level of particulates, as they might do with cell matter or whatever matrices are used, then, and provided you can get good visual representations of that, you can understand what consistency looks like, whatever normal it can be established at, then it's possible then just to look for other types of reactions like color or other forms of density, and you can either do that visually or by using various like optical methods as well, or as I was alluding to earlier, if you want to test the process and show the process doesn't consistently produce particles of concern, is to run a non-cell based solution through the process and then do liquid particle testing at the end of that process.
Okay. Eugene also asks that in adherent cell cultures such as stem cells, would you consider taking a sample from the supernatant or the cell suspension for a sterility test?
That's an interesting question. I would have thought that from the cell suspension it would be very, very difficult to validate the sterility test. It would be far more success, I believe, with the supernatant, just based on the experience of trying to get a meaningful sterility test itself, but of course that's going to be dependent upon the process, the centrifugation process, or the sonication process of releasing microorganisms sufficiently into the supernatant. So that's probably to a degree process specific, but I think I would do what I could to demonstrate release of microorganisms into the supernatant and attempt the sterility test on that.
Okay, I've got a couple of questions about the mycoplasma. Anne Len has asked, can the mycoplasma test be a rapid test in a release testing of GMP product?
Yes, it can do, because there is the general requirements within the European Pharmacopoeia, because that would still fall under the rapid microbiological substitution method. So according to chapter 516 of the European Pharmacopoeia, a compendial method can be replaced by a rapid microbiological method, provided that equivalence or better can be shown. So if you can run the rapid method and the culture-based method in parallel and show equivalency, then it is suitable to do that through typically through a license change application.
Excellent. So a question from Ammon is, how big a problem are mycoplasmas?
Quite a big problem for mycoplasmas are strange. You can go some time without a mycoplasma problem, then get a mycoplasma problem and find it near impossible. Sometimes to get rid of it can result in the loss of an entire cell line, for example, and then result in starting from scratch. It does vary, but it is always going to be a constant problem. So there are some estimates that say that mycoplasma type bacteria outnumber all other types of bacteria by a ratio of 10 to 1. So in terms of what is around us, there's far more mycoplasmas than there are other forms of bacteria. But again, it's a very specific question. You may get facilities who go a very long time without mycoplasma. You're going to get some who are constantly fighting that battle.
Okay, I have a question from Scott, who says, can you comment on how to model media fills or aseptic process simulations related to scale? And do they have to all be done at full scale, or are there reasonable guidelines to be able to downscale the process for this type of testing?
So unfortunately for media fills, so within all the GMPs issued, it directs us back to Annex 1. So it partly depends on the batch size. The annex is obviously, when it goes through the revision, will indicate that time, or we believe Annex 1 will say that process time is more important than number of units, but the time of the media fill should simulate the maximum production time, and then within that there should be a reasonable number of filled units that can replicate the different stages, and where there's need for any manipulations or interventions, that these are adequately covered, and also allowing all of the personnel who will be involved in the process to follow the process. But there's a lot of complexities around media fill design, and that's actually a topic that we haven't actually covered with this webinar series. I think that'd be something to consider. But yeah, media fill design is a whole issue.
Excellent. Okay. Well, we've run out of time now. So thank you to everybody who sent in questions. As I said at the beginning, I'll forward your questions on to Tim and we will circulate them along with the answers, a copy of the slides, and a link to the webinar. Thank you, Tim, for another informative webinar. As we mentioned, this is our penultimate one. The last webinar of the series will be on the 22nd of December, just before Christmas, and it'll be the introduction to mycoplasma and cell product risks. So on behalf of RSSL and the sterile manufacturing team, I'd like to thank Tim again for continuing to deliver such informative webinars for us. And thank you to all of you out there who are taking the time out of your busy days to tune in and listen to us. As I mentioned, please don't hesitate to contact me if you have any questions or if you need any support with any projects. RSSL are here to help you. Thank you. Have a good afternoon.
Thank you.